Welcome back to the Deep Dive, where we like to really kind of get into the nitty-gritty of all things medical microbiology.
Absolutely.
And today we're focusing on a rather interesting bacterium, Francisella tularensis.
Yeah.
And we will be using, as our source material, Principles and Practice of Clinical Bacteriology, second edition by Gillespie and Hockey.
It's a great choice.
And this is a deep dive that is tailored specifically for medical microbiologists.
Yes.
So let's just dive right in. Francisella tularensis, a very interesting organism and kind of a scary one in some ways.
It is, yes. It can be quite a serious pathogen.
Yes.
And it's got that potential as a bioweapon, so.
Right. And some of the reasons for that are, well, it's highly infectious.
Yes.
There's no widely available vaccine.
That's right.
And it can involve a rather lengthy course of antibiotics.
It can indeed.
So let's just start with the basics. What is this organism? What does it look like? How do we describe it?
So Francisella tularensis is a pretty small gram-negative coccobacillus. Okay. So we're talking like a tiny short rod, almost like a sphere, but not quite.
And I know it's a little bit finicky about its environment.
It is, yeah. It's what we call fastidious, so it really needs oxygen to grow. And it also needs this amino acid, cysteine.
I see.
And actually, that's a pretty big clue for us when we're trying to identify it in the lab.
Okay.
That cysteine dependence.
It's not something we see in every bacterium.
No, not at all. Not many gram-negatives rely on cysteine like this.
Okay.
It's kind of unique in that sense, and it also grows very slowly. So,
Yeah.
when we're trying to grow it, it's not like, you just throw it on any old petri dish and it'll be happy.
Right.
We need this special type of agar.
Okay.
It's called cysteine glucose blood agar, but the specific type we use, CHE, is actually recommended by the CDC. And even then, even on this special food, it can take days to grow.
Well, that makes it all the more tricky, I would imagine, when we're talking about diagnosis.
Oh, definitely. Definitely.
Now, you mentioned something that I found kind of fascinating. There are different subspecies of Francisella tularensis.
Right. There are a few, yeah.
And each of those has kind of its own personality in terms of virulence and also where it's found geographically.
Yeah, exactly. So, there's subspecies tularensis, which is also called type A. And that's our real troublemaker. That's the most virulent one. And it's mostly found in North America.
Okay.
And then there's subspecies Holarctica, which we also call type B, which is less virulent, and you find it in more places around the world.
Interesting. So I remember reading about a temperature trick for differentiating it from other bacteria.
Oh, yeah, that's right. So Francisella tularensis, it doesn't really like the heat, grows poorly at 28 degrees Celsius, unlike some other bacteria, like Yersinia pestis.
Oh, okay.
So that can be a really helpful clue for us in the lab.
So it likes the cold weather then?
It seems to prefer it, yeah.
So Francisella, it's a little bit, well, we've profiled our bacterium here.
We have, yeah.
But now let's get into how it causes disease. What's its MO? How does it actually make us sick?
That's where things get kind of interesting because, it's one of the most infectious bacteria we know of. Like, as few as 10 bacteria can get you sick.
Wow.
But it doesn't really have, like, the classic virulence factors.
Okay, what do you mean?
It doesn't make, like, these super potent toxins like some other, nasty bacteria do. And even its LPS, its lipopolysaccharide, it doesn't act like a typical endotoxin.
It's not really setting off our immune system in the way we would expect.
Kind of flying under the radar, yeah. It does have a capsule, which helps it survive, our initial defenses in the bloodstream.
Okay.
But the weird thing is it doesn't actually need that capsule to survive inside macrophages.
Now, macrophages, remind me, those are the...
Yeah, the white blood cells, the kind of like the Pac-Man of our immune system.
Right.
They go around engulfing and, destroying bacteria. But Francisella has this neat trick where it can not only survive inside them, but it actually multiplies in these macrophages.
So it's not just hiding, it's actually using our defenses to its advantage.
Exactly, yeah, turning our own cells against us.
So how does it even get into those macrophages in the first place? Let's talk about the the kind of the doorway that it uses.
Sure, sure. So the most common way people get tularemia, which is the actual disease caused by Francisella, it's through something called ulcer glandular tularemia.
Okay.
And that typically starts with, maybe a tick bite or sometimes a hunter, handling infected meat.
So it's a risk for people who spend a lot of time outdoors.
Definitely. Yeah, definitely.
And then what happens?
Well, after a few days, incubating, you start to get the symptoms and they come on pretty suddenly. Fever, chills, headache.
So pretty much your standard flu-like syndrome.
Yeah, exactly. And then you get this ulcer that forms at the site where the bacteria entered, often on the legs with tick bites. And the lymph nodes, those filters of our immune system, they get swollen and painful.
Ouch.
Yeah, not pleasant at all.
And, how long does it take to recover from something like that?
It can take months, even with treatment.
Wow.
And there are other more severe forms of tularemia as well, like glandular tularemia, which is similar, but you don't get that ulcer. And then there's this really nasty one called typhoidal tularemia.
Typhoidal. That just sounds pretty serious.
It is. That one's it's a systemic infection where the bacteria get into your bloodstream, but you don't have the typical ulcer or swollen lymph nodes, so it can be really hard to diagnose.
Okay.
And if it's caused by that highly virulent type A strain, the mortality rate can be pretty high.
Okay, so we've got tick bites, we've got cuts, abrasions, you mentioned contaminated food or water. But the one that always seems to get to me is pneumonic tularemia.
Right.
That one's pretty scary.
It is, yeah. Pneumonic tularemia, that's when you inhale the bacteria.
Okay.
And sometimes it actually develops as a complication from, other forms of tularemia.
Oh, wow.
And that's where that really low infectious dose becomes especially scary.
Right, less than 10 bacteria.
Exactly, less than 10 through the air, and you can get a full-blown infection.
Wow.
And, the symptoms can be really tricky because they often look like the flu at first.
So you might not even realize how serious it is until it's quite advanced?
Yeah, exactly. And that delay in diagnosis can be, really dangerous.
It's almost like this bacterial ninja, It's lurking in the environment. It can get in through all these different doorways, and it can cause all these different symptoms that look like other things. So how do we even start to track it down?
That's the challenge, isn't it? Isolating Francisella tularensis in the lab, it's not easy.
Oh, really?
And it can actually be dangerous for lab staff if it's not handled properly.
Oh, wow.
So a lot of the time we rely on the clinical presentation, the patient's symptoms,
okay,
and serology.
And serology, that's the study of the antibodies in the blood, right? Looking for the body's immune response.
Exactly, yeah. So we use tests like agglutination and ELISA to look for specific antibodies against Francisella. But there's a bit of a catch here because some strains, they just don't react to the standard antigens we use in these tests.
Oh, so it can be kind of a tricky diagnosis even with those tools.
Yeah, it can be.
Are there any new techniques coming out that are helping us with this?
Yeah, there are some really promising ones, actually. So now we've got ELISA and immunochromatographic assays that can actually detect the bacteria directly in clinical samples.
Oh, so we're actually catching the bacteria in the act, so to speak.
Yeah, kind of like that, yeah. But, those rapid tests, they can be less sensitive than other methods. So often, we need to do confirmatory testing as well.
So there's always a chance that it could still be lurking, even if the first test is negative.
Yeah, that's always a possibility.
So what else do we have in our arsenal?
Well, that's where PCR-based methods come in.
Ah, PCR.
Yeah. PCR stands for polymerase chain reaction, and it basically lets us detect the bacteria's DNA directly.
Okay.
Which is a lot safer than trying to culture it.
Right, right.
And with some of the newer sample processing techniques, PCR can actually be even more sensitive than those traditional methods.
Wow, so we're using cutting-edge DNA technology to track down this bacterial ninja.
Pretty much.
And it's not just about detecting it, right? We can actually use molecular methods to tell the different strains apart.
Exactly. So we've got techniques like REP-PCR, 16S rRNA sequencing, and tandem repeat analysis. All of these help us build a more detailed picture of the bacteria. So we can understand its, unique characteristics, maybe track how it's spreading. That kind of detail is really important for developing effective treatment strategies and, figuring out how to prevent infections.
Well, this is fascinating stuff. We've really gone into the depths of Francisella tularensis here.
Yeah, it's a pretty complex bug.
And it's pretty scary in some ways, too. So I think it's time to move on to how we actually manage this infection.
Sure. Let's talk about treatment.
All right, let's do that. All right, so we've really gotten to know our adversary here, but how do we fight back? What are the best treatments for Francisella tularensis?
Well, when it comes to treatment, aminoglycosides are a go-to.
Okay.
Specifically streptomycin and gentamicin.
Right.
Those are the ones that really, they're bactericidal, so they actually kill the bacteria.
So those are the big guns then when
Yeah.
we're dealing with tularemia?
Yeah, pretty much. Current UK guidelines, they recommend a 10-day course of gentamicin. And it's pretty similar for kids too, but one really important point to emphasize here is that we should never use aminoglycosides as like the only treatment.
Oh, really? Why is that?
Well, especially for acute febrile illnesses,
Okay.
we want to make sure it really is Francisella tularensis,
Yeah.
and we know its sensitivity to the antibiotics before we jump in with the aminoglycosides.
So we want to be sure we're using the right weapon for the battle, so to speak.
Exactly, yeah. So start with a broader spectrum regimen. Then once we have a diagnosis, we can refine it.
Okay. So aminoglycosides, those are kind of the first line, but
Mm-hmm.
what about fluoroquinolones? I know those are becoming more popular, or at least more used for different things.
Yeah, fluoroquinolones are showing a lot of promise for Francisella.
Okay.
They've had some good results. And, they're less toxic than the aminoglycosides, and we can often give them orally.
Oh, okay. That makes it a lot easier for the patient.
Exactly. Much easier to take a pill than, get an injection.
For sure. Are there any specific examples of how well fluoroquinolones have worked against tularemia?
Yeah, actually. There was a recent outbreak in Spain,
Okay.
and they used ciprofloxacin, and it was really effective,
Wow.
even in kids and even in some cases where the infection came back. So it's definitely an option.
That's good to know. So we've got our aminoglycosides, we've got our fluoroquinolones. What about some of the other more commonly used antibiotics like tetracycline or chloramphenicol?
Mm-hmm.
Do those play a role?
So tetracycline and chloramphenicol, those are what we call bacteriostatic.
Okay.
So rather than killing the bacteria outright, they just kind of stop them from growing.
Yeah.
And while they can be effective against Francisella, the problem is they have a high risk of relapse.
Oh, I see. So even if you treat for a long time, it might just come back.
Yeah, exactly. It's like you're hitting the pause button, not the stop button.
Right, right.
As soon as you stop the treatment, the bacteria can just start growing again.
But there are situations, I imagine, where you might have to use chloramphenicol.
Yeah, there's one in particular, actually, meningitis.
Okay.
Because, you see, aminoglycosides don't really get into the cerebrospinal fluid very well.
Yeah, okay.
So if you have a patient with meningitis caused by Francisella, chloramphenicol might be a better choice.
Right, because you need something that can actually reach the infection.
Exactly.
All right. So we've talked about treatment, but, prevention's always better than cure, as they say. What about vaccines? Where are we at with developing a vaccine for Francisella tularensis?
You're absolutely right. Prevention is always the best strategy. And there have been, efforts to develop a vaccine, especially in the former Soviet Union. They actually used attenuated strains of Francisella for vaccination.
Attenuated strain, so that's where you basically take the bacteria and you weaken it.
Exactly, yeah. You make it so it can't cause disease.
Okay.
The most well-known one is called the live vaccine strain, or LVS. And it's actually derived from that less virulent subspecies Holarctica we talked about earlier.
Great.
And, it's shown some pretty promising results in clinical trials.
Oh, that's good.
Yeah, in some cases, it completely protected people. And in other cases, it at least made the illness less severe.
That sounds encouraging. So why isn't it being used more widely? What's the holdup?
There are a couple of concerns. First of all, we don't really understand how the LVS strain is attenuated.
Really? So we know it's weaker, but we don't know why.
Yeah, that's right. It's a bit of a black box.
Okay.
And secondly, it's shown some residual virulence in mice.
Oh, so it could still cause disease in some animals.
Yeah, that's the worry. And because of these concerns, it hasn't been licensed for use in the US or Europe.
I see. So the potential benefits are huge, but we still need more information.
Exactly. We need to be absolutely sure that it's safe before we start giving it to people.
Okay. Well, this has been a really insightful dive into Francisella tularensis. It's a fascinating bacterium, but it's definitely one that we need to take seriously.
Absolutely. It's not something to mess around with.
No. So I think To wrap things up, why don't we test our listeners' knowledge with a few exam questions?
Sounds good to me. Let's see what they've learned.
All right. Okay, listeners, time to see what you've learned. First question.
Yeah.
Which subspecies of Francisella tularensis is the most virulent and mainly found in North America? Think back to our discussion about the different subspecies and where they're found geographically.
Yeah, so this one is pretty straightforward. The answer is subspecies tularensis, which is also called type A.
Right.
And it's the one that causes the most serious problems. Really important to know which subspecies you're dealing with.
Absolutely. Okay, question number 2. What's that key characteristic that makes Francisella tularensis stand out from most other gram-negative bacteria? And little hint, it has to do with what it needs to grow.
It's a bit picky, isn't it? It needs cysteine.
That's right.
Most gram-negatives don't need cysteine to grow, but Francisella tularensis does.
And that helps us identify it in the lab.
Absolutely.
Okay. Question number 3. What's the most severe form of tularemia? The one that's really dangerous, especially if it's inhaled.
The answer is pneumonic tularemia, right? And it can progress very quickly. And with such a low infectious dose, less than 10 bacteria.
It's a little concern.
Yeah, and it can be hard to diagnose at first because the symptoms can look like the flu.
Absolutely. Okay, question number 4. Which antibiotics are the best for treating tularemia? The ones that can actually kill the bacteria.
Our go-to weapons are the aminoglycosides,
Right.
Specifically streptomycin and gentamicin. But it's important to remember what we said about using them appropriately.
Right. Not as the only treatment, especially at first.
Exactly.
And our last question, a bit more challenging. Why isn't the live vaccine strain, or LVS, licensed for use in the U.S. or Europe, even though it's shown some promise in clinical trials?
Well, the LVS vaccine has a couple of issues that are holding it back.
Yeah.
We don't fully understand how it works. We know it's attenuated, but we don't know exactly how that happens.
So there's still some uncertainty about its safety.
Yeah. And it can still cause disease in some animals.
So until we have more information, it's just not safe enough to use widely.
Exactly. Safety has to be our top priority.
Well, that's it for our exam questions. I hope you all did well. We've covered a lot of ground today, exploring this fascinating and sometimes frightening bacterium.
Yeah, it's definitely a bug that deserves our respect.
Absolutely. We've learned about its unique features, how it infects us, the challenges of diagnosing it, and the different ways we can treat it. And of course, we touched on the ongoing search for a vaccine. This is just a small glimpse into the amazing and constantly changing world of medical microbiology.
It's a field that's always evolving. There's always something new to discover.
Well, that's it for this deep dive into Francisella tularensis.
It's been a pleasure.
It's been great having you. And to all of our listeners, thank you for joining us. Stay curious, keep learning, and we'll see you on our next deep dive.